Hello friends, welcome to Biology Made Easy. Today we'll discuss about the nucleosides and nucleotides. Now where these nucleosides and nucleotides are present, you know that in a cell, the nucleus is the most important part that controls everything. In a nucleus, the most important thing is chromosome. In a chromosome, the most important thing is nucleic acids. And these nucleic acids, like DNA and RNA, they are polymers of these nucleotides. That means all the nucleotide or nucleic acids are polymers of nucleotides. Now, if we take one nucleic acid, it will consist of several nucleotides. So, what will be the length of the nucleic acid? That depends upon the number of nucleotides. For example, in 5X174, one virus, there is 5,386 nucleotides, while in a lambda phase, there are 48,502 base pairs. Here I have written nucleotides, here base pairs, because this is single stranded, this is double stranded. Similarly, in E. coli, 4.6 into 10 to the power 6 base pairs. In human being, in a diploid cell, 6.6 into 10 to the power 9 base pairs. So, the length of the nucleus depends upon the number of nucleotides. Now, each nucleotide again consists of three things. It consists of one nitrogen base, one pentose sugar, and one phosphoric acid. This phosphoric acid is in three sides there will be hydroxyl groups, and in one side there is one oxygen, H3PO4. Now, nitrogen base and pentose sugar, when these two will combine together, it will give rise to a nucleoside, the first one. And when a phosphoric acid will be attached to it, it will give rise to a nucleotide. Now, the pentose sugar present in nucleic acids is again of two types. It may be a ribose sugar present in RNA and deoxyribose sugar present in DNA. So, what is the structure of this ribose sugar? These are pentose sugars. This is number 1 position, position number 2, 3, 4, and this is 5. Here there will be one OH, H, OH, here H, and here CH2OH. This is the structure of a ribose sugar, which is pentose sugar. Now you see, the hydroxyl groups are present in position 1, 2, 3, and 5. But when it will give rise to deoxyribose sugar, when this oxygen present in the second position will be removed, that means on either side will be H and H, it is a deoxyribose sugar. That is why sometimes question is asked, the deoxyribose sugar differs from ribose sugar in what respects? So there is absence of oxygen in position 2. And this also makes a major difference between RNA and DNA, because as we will discuss in the coming lectures, you will find that the OH present in first position will be utilized, third position will be utilized, fifth position will be utilized, but the second position OH group will be not utilized. That is why in case of RNA, this OH group will be there. That is why RNA is catalytic, but the DNA is not present, so it is not catalytic. This is a very important question. Now you come to the nitrogen base. The nitrogen bases are again of two types. They may be purines or they may be pyrimidines. The purines are double ring structures while pyrimidines are single ring structures. Now examples of purines are adenine and guanine, and examples of pyrimidines are thymine, cytosine and uracil. In case of a DNA, there is adenine, guanine, thymine and cytosine, while in case of RNA, there is adenine, guanine, cytosine and uracil. Thymine will be absent. Now, the purines are double ring structures. So double ring structures means they will look like this. This is position number 1, 2, 3, 4, 5, 6, 7, 8 and 9. And the nitrogen bases are present 7, 8, 9. Correct. And nitrogen bases are present at the position 1, 3, 7, 9. 1, 3, 7, 9. At these positions only nitrogens are present. Similarly, in case of pyrimidines, which are single structures, 1, 2, 3, 4, 5, 6, and nitrogen bases are present only at position 1 and 3. Now, in these different nitrogen bases, adenine, guanine, thymine, cytosine, uracil, at different positions there will be amino and oxo groups. So, adenine is known as 6 amino purine. That means amino group is present at the 6th position in case of adenine. Guanine is known as 2 amino purine. That means the amino group will be present at 2nd position. Then cytosine. Cytosine is known as 6 amino 2 oxopyrimidine. So here in case of cytosine you will find that at the 6th position amino group is there, and at 2 position there is oxo group. In case of thymine, 5-methyl-2,6-dioxopyrimidine. In the 5th position, there will be one methyl group. Here there will be one methyl group, and at the 2nd position and the 6th position you will find oxo groups are there. In case of uracil, it is 2,6-dioxopyrimidine. Then only in at the position of 2 and 6 there will be oxo groups. Again you will mark there is a difference between thymine and uracil. The only difference lies at the 5th position of thymine. There is a methyl group which is absent in case of uracil. That is why thymine you can call at 5-methyl-uracil. So these are the basic ideas about the nitrogen bases, sugars and phosphoric acid. Now, how they will give rise to nucleosides and nucleotides, let us discuss. This is one ribose sugar. And this is one nitrogen base. This is 1, 2, 3, 4, 5, 6 positions are there. And in this pyrimidine, which is single structure, nitrogen present at position 1 and 3. So here it will be NH at position 1. Now, the hydroxyl group present in position 1 of the ribose sugar, it will combine with the hydrogen at position present at 1, and it will give rise to one glycosidic bond, which is a covalent bond. That means the pyrimidines, they are attached to the sugars by means of 1,1 N-glycosidic bond. That means the position of 1 of pentose sugar and position 1 of pyrimidine. But if it is a purine which is a double ring structure, in that case this is the sugar, and this is the OH group. In this case, it is a double ring structure. So here this is the ninth position, and the ninth position there is a NH group. So this OH and this H will combine together to form a glycosidic bond. So in this case, it is known as 1,9 N-glycosidic bond. These two are very important for examination point of view. So now you see, the ribose sugar is attached with a pyrimidine. It gives rise to a nucleoside, because when nitrogen base is attached with sugar, it gives rise to a nucleoside. Now, if this is ribose sugar, then this will be a nucleoside for RNA. If it will be deoxyribose sugar, then it will be a nucleoside for DNA. Now, the nucleosides of RNA and DNA will have different names. Suppose you take the example of RNA. So in RNA, if it is adenine, then the name of the nucleoside will be adenosine. Guanine, guanosine. Cytosine, cytidine. That means adenosine, guanosine, cytidine. These are the names for different types of nucleosides of RNA. But if it will be DNA, in DNA, the ribose sugar is replaced by deoxyribose sugar, so the names will slightly change. In case of DNA, it will be deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. In case of RNA, thymine is absent, it is uracil, so it is known as uridine. You correct that one? So there are 4 different types of nucleosides for RNA: adenosine, guanosine, cytidine, and uridine. And for DNA, the 4 are deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. Now, how the nucleotides are formed? That is very interesting. Now, already nucleosides are formed. Now, how the nucleotide is formed? Now, the phosphoric acid, this is the 5th position, and the phosphoric acid will be attached to this 5th position. This is phosphorus, this is one hydroxyl group, this is one hydroxyl group, this is one hydroxyl group, and this side one double bond O is there. Now, the hydroxyl group of this CH2OH will combine with the hydrogen of the phosphoric acid, and here one molecule of water will be liberated. Now, what happens you see? Here there is phosphate, then there is oxygen, and here there is C. This bond is known as phosphodiester bond. And now the nucleoside is converted to a nucleotide. So in a ribose sugar, at the first position the nitrogen bases are attached, and in a nucleotide the phosphoric acid is attached to the 5th position. That means you see, at the first position and the 5th position the OH groups are now utilized, only at the 3rd position it remains. So now this ester bond, and now it's a nucleotide. So now what are the different types of nucleotides for RNA and DNA? So for RNA, you can call it adenylic acid, because the acid is attached, adenylic acid or adenosine monophosphate. Then guanylic acid or guanosine monophosphate, cytidinic acid or cytidine monophosphate, uridylic acid or uridine monophosphate. For DNA, deoxyadenylic acid or deoxyadenosine monophosphate. Then deoxyguanylic acid or guanosine monophosphate, deoxythioadenylic acid or deoxythioadenylic monophosphate, and deoxycytidelic acid or deoxycytidelic monophosphate. This is the difference. Only deoxy will be added in case of DNA. So in this way, the nucleotides and nucleosides are formed. But how it will give rise to a polynucleotide? That is again more important. Now see, nucleotides, different types of nucleotides are formed, but how a dinucleotide and a polynucleotide is formed? So this is one nucleotide, this is another nucleotide. In this nucleotide, here also you will find CH2OH, this because this is the fifth position, this is the fourth position, third position, this is the second position, and to first position is already attached. So here also you will find one phosphate group is attached. So here OH, P, double OH, double bond O, here OH there. And here in this third position also, this side is H, this side is OH. Now you see the OH of this and the H of phosphoric acid, it will liberate one molecule of water. Now what is happening you see. You mark in detail. Here what is there? C. Then after C next what is coming? O. Then next what is coming? P. Then next what is coming? O. Then next here what is coming? C. That means this is COP is one phosphodiester bond, and again another COP, this is another phosphodiester bond. That means on either side of this phosphoric acid, on this side one phosphodiester bond is there, and then the other side one phosphodiester bond is there. That means these two nucleotides are attached by means of phosphodiester bond. At this point, most of the students confuse what is ester bond and what is diester bond, and where we will use it. So I hope now it is clear to you. Now this phosphodiester bond, how will I name it? Now you see, this is the third position of this nucleotide, and this is the fifth position of this nucleotide. And this phosphodiester bond, it is usually catalyzed by the enzyme DNA polymerase. And the DNA polymerase will catalyze this reaction only in this 3' to 5' direction. That is why this phosphodiester bond is known as 3' to 5' phosphodiester bond. That is very important from examination point of view. Now, if a third nucleotide will be attached, where it will be attached? Whether to this side or to that side. Again, that is important. That will be attached only to this side, never to that side. When a third nucleotide will come, here there is OH, and again we will get phosphoric acid, and it will continue the direction. That means now the nucleic acid will run in this direction. And in the first nucleotide, there is this is the 5' end, which terminates with the phosphate. And when we will go on dinucleotide, tri and polynucleotide, at the final end there will be 3', which will end with the sugar. That is why all nucleic acids, they run only from 5' to 3', never in the reverse direction, because both DNA polymerase and RNA polymerase, they catalyze only in this direction. This is very, 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 very important. So this is the idea regarding the nucleosides and nucleotides. Now, what are the importance of these nucleotides? That is again very important. Very few students know about it. First thing, the nucleotides, they are the monomers of nucleic acids. This is the most important thing. Second thing, the nucleotides, they give rise to energy carriers like ATP, GTP, CTP and UTP. Adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, uridine triphosphate. That means when to one nucleotide, another two phosphate molecules are added, they become energy carriers or energy currencies of the cell. So from this you can mark whether ATP is more nearer to RNA or DNA. From the full form you can guess it. Adenosine, and adenosine belongs to RNA. If it would have been DNA, then it would have been deoxyadenosine. So this is more related to ribonucleic acid. The third one, chemical messenger. You have heard the name of cyclic AMP, which is chemical messenger present in cell, and that is also a nucleotide. And the fourth one is vitamins. You have heard the names of NAD, NADP, FMN, FAD, all these things. So suppose take the case of NAD, nicotinamide adenine dinucleotide. Means two nucleotides are joined together. How? Nicotinamide, sugar, phosphate, sugar, adenine. So you see this is one nucleotide, this is another nucleotide. It becomes a nucleotide. So these are the important functions of nucleotides. I hope you have understood it perfectly, because this is the basic idea in the molecular inheritance chapter. And without a clear understanding about this, it will be very difficult on your part to understand other things. Now, I will give you a few questions. Number one question. Cytidine is present in RNA, DNA, both, none. One second question. In a nucleotide, phosphate group is attached to position dash of sugar. A choice 1, B choice 2, third choice 3, and fourth choice 5. Third question. Thymine is attached to sugar by forming A choice 1 glycosidic bond, B choice 9 glycosidic bond, C choice 3 glycosidic bond, D choice 7 glycosidic bond. You will be able to answer all these questions. So thank you very much for watching it, and I wish you a very good day.